All right, so we're back with covering the structure of DNA. Really, the goal is to get across what exactly does this double helix look like? What is it made of? How does everything fit together? And so, just kind of a really quick FAQ about the DNA molecule. It stands for deoxyribonucleic acid. So it falls into the macromolecules of nucleic acids, which deal with genetic information. The deoxyribose has to do with the sugar. So the sugar that we find in DNA is deoxyribose. It contains two strands that twist, forming a double helix, and the function of it is to store our genetic information. And so, looking at this DNA molecule, if we break it down into the smaller pieces, we know that DNA is made up of many repeating units called monomers. And these units are specifically known as nucleotides. So these nucleotides are assembled to make our DNA molecule, or the DNA that is in our chromosomes. That is made up of three different parts. It is made up of phosphate group, which if you remember from first semester, I call it the balloon. It is made up of a sugar, which I call a house. And it is made up of your nitrogen base, which I called the garage. So you see that here. So this is just one nucleotide. That base could be adenine, guanine, cytosine, or thymine. And then the other side of that molecule would be its matching pair complement. There are four different bases. You have your purines, which are double, and pyrimidines, which are single ring structures. Um, how I remember these is pyrimidine has a Y in it, and I think of a pyramid as a single triangle or single thing. Um, and so pyrimidine has a Y in it, cytosine has a Y in it, and thymine has a Y in it. And if you remember back to the history, this all comes from Chargaff's rule. And Chargaff was able to discover that A pairs with T and C pairs with G. Putting this to practice, if you really take a quick second here, given the sequence below, give me the complement. And this is something that you will have to do when it comes time to take a quiz, when it comes time to look into RNA and protein synthesis. So I'll try to pencil out a few of them. So you'd have T, A, it's a bad A, but we'll go with it. A, and go on and go on. And I'll give you about 10 seconds or so, or 15 seconds to knock that out. So looking at the strand ATT, GC, AT, GAA, GGC, you should be able to produce the complement, the opposite side, that is TAA, CG, TAC, TT, CCG. This is something that you'll have to do. Looking at the double helix, the nucleotides are arranged in a shape known as double helix or a twisted ladder. Couple questions that you see here is how many nucleotides do you see? And if you're going to look at the nucleotides, you have to look at each one of these little phosphate sugar base combinations. So in this example, we have 1, 2, 3, 4, 5, 6, 7. That's a terrible 8. But in total, we have 8 nucleotides. You could also say we have 4 base pairs. If you remember from the last lecture, the person responsible for this was Watson and Crick. A few more facts about this. You have the backbone, or the sides of the ladder, that is made up of sugar and phosphate. Those are made up of really, really, really strong bonds. They're called strong covalent bonds. I call them phosphate bonds, but they're not easily broken. We don't want our DNA to be damaged, so we need these to basically protect the bases in the order of the bases that we have. The nitrogen bases make up the steps. These are connected by weak hydrogen bonds. It makes it easier to break so that we can open it up, transcribe it, which means rewrite it or copy it. And then in the end, the order of the bases tells me what we're going to make. DNA is the instruction for life. In the end, it's the instruction for proteins, which we'll get to next chapter. But before we move on, looking at the hydrogen bonds that exist, there are two things I want to point out. Between guanine and cytosine, you should see three little bonds there. It's because there are three hydrogen bonds. It makes it a little bit stronger than the two bonds that exist between adenine and thymine. And so a question that usually pops up on a quiz is which bases are easier to break? The bonds between G and C or A and T? And it's A and T because there are only two bonds that need to be broken. The other thing that I want to point out is the term antiparallel. And if you remember doing the model, you remember that I said DNA is antiparallel, which means it runs in opposite directions like a two-way street. So if you see here, the O's that are in my deoxyribose, which means it has oxygen in it, on the left-hand side, the O's are pointed up. So that strand runs, we'll say up, and on the right-hand side, the O's are pointed down. And so that is what antiparallel means, that they run in two opposite directions. And that is pretty much it for the basic structure. We're going to do some color notes in class to help solidify some of these terms, what the sugar and phosphates are, and what the bases are. And so we'll go from there later on this week.